Today we can study cellular junction, and cellular junction is nothing but cells and the junction between them. So it can be a lot of types, and we're gonna talk about majorly 7 types. First junction is tight junction. As you can see, it's tight. Other one is zona adherence. So this junction is that zona adherence and desmosomes. So these are actually partially connected. Cool. Gap junction. They have gaps inside. Why we need gap? Because we need fast transport. Cell basal lamina. Cell. This is the base layer connection. Cell basal lamina. Hemidesmosomes and focal adherence. We will talk all of them. We'll talk about all of them. Cool. Let's talk first about tight junctions. They are tight junction, and they are also called zona occludens. And the word root of occludens is quite amazing. Oc means towards, and cludens means to close. So the one that closes off. So it's a nice name, honestly. Cool. Tight junction. Seen where? In GIT, in blood-brain barrier, in renal. So we don't want to lose any sort of substance, or we don't want any sort of communication, fast communication between two cells. So that's why we want GIT. We want blood-brain barrier. We don't want things leaking into your brain. So that's why we have blood-brain barrier. And just imagine if you have, bad junction like towards the cell, or bad junction between your cell. Just imagine a lot of toxins leaking from your blood into your into your brain. Is that good? No, it's not good. Cool. We don't want that. They have two proteins, occludens and clodens. So both DINs. Cool. Now you will remember tight junctions. Now if they are mutated, there will be renal impairment, or there can be problems at triple B, or there can be problems at GIT. Cool. But if this clodens are messed up, you will have renal impairment. And what will happen? Your body will lose a lot of calcium out of your body. Your body will lose a lot of calcium, and the magnesium levels will be low as well. So this will be hypomagnesemia and hypercalciuria in the renal, and this will affect as renal stones. So if someone has, someone in particular, has a lot of renal stone happening again and again, that can be indication that his clodens may be not working. Okay, so now we understand tight junctions. Again, tight junctions, cells like this. So it means it's tight. No communication like this, nothing. So what happens is that two cells like this, the nutrition or the substance moves across paracellularly. So paracellular movement of the substance because it's tight. Cool, we understand it now. Okay, we are done with tight junctions. Let's see zona adherence. So adherence means they are stick with each other, and zona means zone. That's it. So zona adherence and desmosomes, they both are called cell adhesive junction. Cool. These cells are partially attached with each other. Adhesive, it means they are attached with each other. Adherents and desmosomes. So in adherence, we have CAMs, CAMs. What is CAMs? Cell adhesive molecules. We need CAMs. Now what are CAMs? Cadherins. Do you remember E-cadherins from previous lesson? That's what I was talking about. We have integrants and we have selectants. We need them. Why we need them? Because in your plasma, in your hemat physiology, you will study about integrants and selectants, how plasma levels are, and how the coagulation thing works. Cool. We don't want to hear them right now. So CAMs, we need cell adhesive molecules. And for cell adhesive molecule, there are 3 types of cell adhesive molecules: cadherins, integrants, and selectants. In selectants, okay, cool. Now we understand this. Let's study the structure of a basic adherin or a basic desmosome. Now note that desmosome might have different name. of the part, okay? But the structure remains the same. Primary protein remains as a primary protein. They may be named as, maybe, I don't know, they in desmosomes, I don't remember the names of these things, but I should remember, but I don't remember. So you can say that the structure more or less remains the same. So in this structure we can see there is one primary protein, then there is linker protein, and then there is the cytoskeleton itself. Cool. The cytoskeleton and primary protein is connected via linker protein. That's it. Now the name of these proteins can change depending upon if it is a atherin or if it's a desmosome. Cool, now you understand that. So desmosomes are cell adhesive junctions. Cool. They are present at the part of mechanical stress. Where is mechanical stress? Heart, skin, uterus, cervix. These both contraction. Heart as well, contraction and relaxation. Skin, mechanical beating from your bully. Cool. The skin goes under mechanical, stress. And that's why we have desmosomes. Okay. Now, if the skin has antibody from itself towards its own desmosomes, for example, antibody for desmoglin. Again, I told you the name of these primary protein, these things can change. So, antibody for desmoglin, we call this pemphigus disorders. There are many pemphigus disorders. Cool. Gap junction. There is gap between two cells. Gap junction. Cytoplasmic molecule move from one cell to another via the gaps, and these gaps are really fast. So the speed I'm talking about, okay, this is the gap, okay, 3 to 4 mm gap. That's it. They are like close, really close, 3 to 4 mm. This is rapid communication. Seen where we need rapid communication. We need rapid communication in cardiac muscles. We need them in neurons. We need them in nerve fibers. Okay, what else? So the protein found in gap junction is connexon. Okay, if connexon is damaged, so what kind of connexon can be damaged? Connexon number 32, connexon number 40 can be damaged, and if 32 is damaged, it's called Charcot-Marie tooth. It's the neurons that's damaged. If connexon 40 is damaged, I'm talking about cardiac failure, idiopathic atrial fibrillation. So if your 32 is damaged, that's neuro. If 40 is damaged, that's cardio. So, okay, remember this. I don't know how to remember this. 32 stands for neuro. Okay. Cell basal lamina. Cell basal lamina. Cool. This is a junction. If they are disrupted, the cell starts moving. So they are together in such a way that if they are disrupted, the cell start to move from one place to another, and that's how the cell is moving. And some people, they call this kind of movement metastasis, and that's how the cancer cell cancer cell, that's how metastasis is done. You get me, right? Cool. Hemi is half, focal is also like focused. So these two are quite similar in nature, and what happens is that this adhesion is attached to actin. Do you remember actin? We just talked about actin. Microfilaments, smaller in size, actin and myosin, too. Like this, if you remember this. Okay, so focal adhesions have actins, and hemidesmosomes are attached to intermediate filaments. Where were they? Oh, remember this? Cool. Okay, so that's the only difference between these two. So now we know cellular junction. Let's talk about hormones as well. If we are like, let's talk about hormones. So hormones can be classified upon amino acids, peptide and protein derivatives, cholesterol, steroids, and vitamins. So notice that I have divided them into four types. uh Wait, so these two, these are proteins, and these are fats, majorly lipid soluble, basically soluble. So these are lipid soluble and these are fats. So if something is lipid soluble, it means it can cross the membrane, right? It can cross the this membrane, this bilipid layer membrane, right? So cholesterol derivatives such as estrogen inside your woman, testosterone inside your man, aldosterone, cortisol, the stress one, they can cross the lipid. So it means that their receptors are inside the cell. Cool. For example, vitamins A and D. Cool. These two can cross as well. So their receptors must be inside. Okay. Amino acid derivatives such as tyrosine, tryptophan, histidine, tyrosine, epinephrine, norepinephrine, dopamine, T3 and T4, they cannot cross the barrier. They cannot cross the bilipid layer. Tryptophan, such as serotonin and melatonin, they cannot cross as well. Histidine, that is histamine, it cannot cross the membrane as well. Let's talk about peptide and protein derivatives or hormones. For example, thyrotropin releasing hormone, TRH. This is 3 amino acid hormone, so it's a smaller one. But insulin, on the other hand, is bigger one. It's 51 amino acids. And PTH, the parathyroid hormone, is 84 amino acids. I usually like to call PTH, this parathyroid hormone, as potassium thrashing hormone, because it throws the potassium out of your body, and it also reabsorbs a lot of calcium from your bones. So okay, this is that, it's we don't need to know that right now. So parathyroid hormone 84, big in size, not so big in size. Cool. Okay, let's talk about receptors. We can have extracellular, we can have intracellular, or we can have neurotransmitter. Let's talk about extracellular first. Cool. Before really like studying about the receptors, we should really understand about the concept of first messenger and second messenger. I make this cell. This is a cell. Cool. This is a nice cell. There is a hormone. He wants to talk to this cell. This is a nucleus. This is where the cell talks. This hormone cannot go inside. Why? Because it's a protein. It can be norepinephrine or epinephrine. It cannot go inside. So this hormone must need someone to talk to the cell. Cool. Something inside. Cool. This will go here at the membrane and talk, and then something from the inside goes to the nucleus and then talks. So this inside player is called second messenger, and this outside player is first messenger. Cool. Now you understand first and second messenger. Okay, the first type of extracellular, it means the cell is here. It's extra, not intra. If it's intra, it means here. It's extra, it means it's on the membrane. Okay, it's between, it's transmembrane. Okay, it's passing the membrane. Okay, extracellular. So GPCR, G protein coupled receptors. Cool, let's talk about them first. So GPCR, G protein coupled receptors. They are 7 pass transmembrane or serpentine receptors. They pass 7 times through the membrane. They have 3 subunits: alpha, beta, and gamma. Cool. What these subunits do? They perform signaling usually. They perform the pathway. They are used in pathway. I will tell you what happens. So together they are alpha, beta, and gamma. Cool. But hormone binds. Okay. But why did I write but? I don't know why did I write but. Okay. So hormone binds. Where the hormone binds? Hormone binds at this receptor. outside the cell. Cool. And this receptor is together with this subunit, okay? And then hormone binds, and then sulfa, this subunit, separates. Why it separates? Why not beta and gamma? Because beta and gamma form more stable dimer together, and beta gamma are together almost all the time. They're like a couple. They act as one unit. If they're separated, they're non-functional. Cool. So what happens? So alpha unit has GTPase as well itself. So it acts on GTP and then converts this GTP into GDP with the help of GTPase. Now what this GTPase does? It phosphorylate GTP into GDP. So this is duo. This is tri. It removes one phosphate, and this phosphate is how the energy moves inside the cell. Cool. GDP. Now in this process, energy liberated, and then energy goes downstream reaction. What is second messenger? Cool. We need messenger. What is second messenger? Something that is inside the cell. It can be camp, it can be calcium, it can be cyclic GMP, cyclic AMP, calcium. Okay, all second messenger, they activate protein kinase ultimately. So all the second messengers have the work of activating protein kinase ultimately. So now GPCR, that is G protein coupled receptor, use any subunit. Okay, it can be alpha, but usually it's alpha. Cool. So they use alpha subunit, and what this alpha subunit does is that if a hormone is activated, I'm talking about what happens after this. I want to tell you now. So what happens is that if this alpha subunit activates GS protein or GI or maybe GQ or maybe GT, what happens? If it activates GS, S stands for stimulatory, I stands for inhibitory, I don't remember Q, but T stands for transduction. Cool. S, something is stimulated, I, something is stopped, Q for calcium, remember, T is for transduction. I will tell you what. So what happens is that the hormone is activated. If a hormone activates GS, this stimulation increases the level of adenyl cyclase AC, and then this results in increased CAMP activity, and then later the CAMP activity can result in different outcomes. Okay, if GI is activated, it means inhibition, so the level of CAMP goes down. Okay, if GQ is activated, then what happens? The level of phospholipase C, important, increases, and C stands for calcium and C stands for phospholipase C, so level of calcium increases. Cool. GT, I made this look like an I, so GT is for transducin. Trans means across, ducin means to lead, so a protein that leads across. Now to lead across we need eyes. That's why we see them in retina, especially in roads. Okay, cool. So rods and cones, there are two types of cells present in retina. So at the rods we have GT, and if they're activated, so what happens is that it activates phosphodiesterase PDE, and this reduces cyclic GMP. Cool. Now we know this. Okay, so now we understand second messenger. Let's talk about CAMP, let's talk about calcium, and let's talk about cyclic GMP. Okay, let's first talk about CAMP. CAMP uses protein kinase, and protein kinase, this is what happens after CAMP. So CAMP specifically uses protein kinase, and this protein kinase actually adds protein to ATP, and then ATP goes through phosphorylation. Not ATP goes through phosphorylation. This phosphorylation of protein happens. The PO4 is attached to protein, and this ATP turns into ADP after the CAMP level rises. So any hormone that need CAMP is Any hormone that need camp are hormones of metabolism, hormones of electrolyte secretion, and water reabsorptions. So metabolism, glycogeoneolysis, glycogeoneolysis. So which hormone? Glucagon. Glucose is gone. We didn't eat for a lot of time. And epinephrine works on camp level increases, and then this process, glycogenolysis, happen. Cool. Glycogenolysis happen. Okay, let's talk about secretin. This is the first hormone discovered, and this secretin releases bicarbonate, and this bicarbonate is mixed into pancreatic juice, and then this juice is a natural antacid. Cool. Water reabsorption via V2 receptor. V stands for V2 and V stands for vasopressin hormone. So water reabsorption, we don't want to lose water in desert, right? So V2, it stands for water because H2O also has 2. I mean, that's how I remember. V2H2O, 222. Okay. So via V2 receptor, the vasopressin activates V2 receptor, and then camp level increases, and then camp level increases aquaporin 2. This is also one hormone. Aquaporin 2 results in water resorptions. Cool. Now imagine this secretin pathway, this electrolyte secretion pathway, is hijacked by some toxin. Cool. Let's talk about cholera toxin first. So the cholera toxin stimulates GSS, stimulatory. Camp level increases, electrolyte secretion increases, so diarrhea happens, and ultimately you can die. What about pertussis? Pertussis inhibits GI, so it's inhibiting the inhibitory system. So it means the camp level increases because you are stopping the system that stops. So you're allowing, actually, you're increasing the camp levels. So if you increase the camp level, you will lose again a lot of electrolytes. If you lose a lot of electrolytes, where will you lose it? You lose it in sputum. So sputum loses electrolyte during cough, and we call this whooping cough. So it's very, very classic. Cool. What about if the GS, is the GS alpha unit is mutated, and it locks it on, it's on, and adenylate cyclase, it's also on without ligand. Cool. What will happen? The camp level increases dangerously, and then what will happen? Puberty, precocious puberty, skeleton problems, and something that is called as caffeine loss spots, and we call this McCune-Albright syndrome. This will happen if your GS alpha unit is mutated. So guys, please don't mutate your GS alpha units. Okay, we won't mutate our GS alpha units. Cool. Let's talk about the other messenger, the second messenger, that is calcium. Okay, so hormone that uses contractions. So the place where contractions happens, constrictions happen, and exocytosis happen. Anything that's coming out of the cell, we need calcium. C stands for calcium, C stands for contraction and constriction. Okay, contraction of what? Constriction of uterus. Which hormone? Oxytocin. Constriction of what? Constriction of blood vessel. Via what? V1. So V1, it is vasopressin, and V2 was for water reabsorption, because 2 and 1 is, I don't know, constriction, vasopressin. And it's such a good name, angiotensin hormone, because it tenses, it constricts the blood vessel, increases the blood pressure. And exocytosis, it means something is getting outside of the cell. So what is it? TRH and CCK. Cool. Both hormones releasing hormones, they both are peptides, TRH and CCK. We just read about them here. Protein and peptide. Thyrotropin releasing hormone, TRH. It's a three amino acid chain. Okay, cool. Let's study about cyclic GMP. Remember two. Remember the two NS. I will tell you why. Relax and vasodilation. Cyclic GMP. Cyclic this cyclic AMP. Cyclic AMP for metabolism, for electrolyte, and for water reabsorption. For calcium, constriction, contraction, and exocytosis. So basically, cyclic GMP is opposite of calcium. So contracts, relaxes. So cyclic GMP works via nitric oxide hormone and natric uretic peptide, NUP. And for example, there is one hormone called atrial natric uretic peptide, ANUP. This hormone is secreted by atrial myocytes because of the stretching of them. Okay, when these myocytes are stretched because of high blood pressure or any sort of high turbulence, this hormone called natric uretic peptide is released, and it releases and it results in vasodilation and relaxation of the capillary. Cool. Let's talk about tyrosine kinase receptor. Okay, why are we talking about tyrosine? Why are we talking about tyrosine kinase receptor? Because we are studying extracellular receptors. So we already studied GPCRs. Let's talk about tyrosine kinase, Janus, and serine. Cool. Tyrosine kinase, something with insulin, because I Janus kinase, beautiful name of a beautiful girl, Janus. We will talk. And serene's pachiti, almost like sex. We will talk serene and sex. Okay. Tyrosine kinase. Remember the I. The I stands for insulin, needed for cellular growth and differentiation. Insulin needed for cellular growth and differentiation. You know that? Clinically. Okay. So if this receptor is messed up, what's going to happen? There's something called as BCR-ABL gene. It's a fusion gene, and it's found in Philadelphia chromosome, and it activates tyrosine kinase, and the growth is going up. If you have this gene, then the growth via tyrosine kinase is amazing, and it's so amazing that you end up with something called as chronic myeloid leukemia. So what happens is that you have BCR-ABL, and then you have Philadelphia chromosome, and then you have tyrosine kinase, and the growth is amazing, and then you have CML, and for treatment you need something called as imatinib, right? Imatinib. And what this does is that it stops tyrosine kinase. I think that it tries to block tyrosine kinase. Cool, now you know tyrosine kinase. Let's study about Janus. Janus is a beautiful girl name. Maybe it's a boy name, I don't know. So this Janus kinase receptor, also known as cytokine receptor, because a lot of cytokine from your body, a lot of inflammatory messenger cytokines from your body attached to it. So this is present where? It's present on the GH. It's present. What attaches to Janus kinase? GH hormone, prolactin, erythropoietin, and leptin. Cool. How to remember this? Giant holds a milk bottle in a pool of blood, trying to eat and drink. The name of this giant is Janus. It's a girl. Cool. The giant GH leads to giantism. Prolactin is for milk, erythropoietin is for blood, and leptin is for hunger. Cool. There's something called as JAK, that is Janus kinase, and there's something called as STAT, which signal transduction activator of transcription. So what happens is that JAK is inside, and then STAT actually phosphorylate, and the STAT is like this, okay? And then after it's phosphorylate, it becomes like this with PO4, okay? And then this is how the energy is transferred, and then ATP becomes into ADP, and this 1P, this triphosphate, and this 1P from the triphosphate stuck with STAT and then STAT then goes to nucleus for transduction and activation of transcription. Cool, new genes formed. That's how the genus works. Let's talk about serine. Serine kinase, sexy name, I must say. So why sexy? It's sexy because it's used in reproduction. And serine kinase have two activins and anti-Mullerian hormones. Okay, what is anti-Mullerian hormone? Mullerian hormone. So sex differentiation happens in boys. If this hormone is not present in men or boy, there's something called as Mullerian duct, and they will eventually have ovaries. Because Mullerian duct become ovary, but because of anti-Mullerian hormone, this Mullerian duct eventually converts into male reproductive organs. Cool, now you understand this. Why we need activins? We need activins to activate FSH release from anti-pituitary. We need FSH for good period cycle. FSH is follicle stimulatory hormone, so it stimulates that follicle that you have if you are a woman.